Good afternoon, everyone. I am Dr. Richa from MLT Department of Avilash University. I would like to express my sincere gratitude to Avilash University and NDLI Club for giving me this wonderful opportunity to present a presentation. Today, I am going to talk about a fascinating topic, bacteriophages, unlocking new applications. Bacteriophages are found everywhere in nature, in water, soil, and even inside human body. In this presentation, we will discuss about the introduction, history, characteristics of bacteriophages, structure of bacteriophage, life cycle of phages, and various application of phages in various fields. Bacteriophages has vast application in medical sciences and in scientific research. Can you imagine? Even bacteria can get a virus. The virus that infect and replicate within a bacteria and archaea are called bacteriophages. Bacteriophages are ubiquitous viruses and found everywhere where bacteria exist. Bacteriophages are the most abundant biological entity on the planet, more than every other organism on earth, including bacteria. In this slide, we will discuss about the history of bacteriophages. In 1915, British bacteriologist Frederick Todd discovered a small agent that infect and kill bacteria. In 1917, French Canadian microbiologist Felix de Herle on 3rd September 1917 discovered an invisible antagonistic microbe. De Herle called the virus a bacteriophage, means a bacterium eater, based on its ability to destroy bacterial cells. He successfully used bacteriophages to treat dysentery patients. His discovery paved the way for using bacteriophages as tool in microbiology and genetics. In 1952, especially the work of Hershey's as contributor to the Hershey cheese experiment, provide the evidence with the help of phages that its DNA, not protein, was the genetic material of life. Hershey and Chase used T2 bacteriophages because bacteriophages are simple in structure and made up of only DNA and protein. This makes them ideal for determining which molecules carry the genetic information. In 1969, Max Delbruck, Alfred Hershey, and Salvador Lucia were awarded the Nobel Prize in Medicine for their discoveries of replication of viruses and their genetic structure. In 1896, Ernest Hankin reported that something in the water of Ganga in India had a marked antibacterial action against cholera, and it could pass through a very fine porcelain filter. Hankin observed that people who drank water from the Ganga and Yamuna seemed to have lower rate of cholera infection. His experiments show that cholera bacteria died even when exposed to Ganga water. Even diluted water of Ganga can kill the cholera infections. In this slide, we will discuss about the characteristics of the bacteriophages. Bacteriophages are highly specific to particular bacterial species due to receptor recognition. Some phages infect only a single bacterial species, while others have a broader host range. Bacteriophages specifically infect bacteria. They are filterable through filter and are sensitive to heat. In next slide shows the structure of bacteriophages. Bacteriophages have diverse structural form. A common form consisting of a head tail, tail fiber, and base plate. The best known example of phage is the T4 bacteriophage, which infect E. coli. The main structural component of bacteriophage is head or capsid. Capsid is a protein shell made up of small units of capsomeres that encloses the phage's genetic material. Capsid may be capsid icosahedral, spherical, filamentous, rod-shaped, or complex in structure. Capsid contain nucleic acid, DNA, or RNA, which can be either single strand or double strand. After capsid, collar connects the head to the tail. In some phases, it helps to stabilize the structure of bacteriophage. Tail is a tube-like structure extending from the head. It functions as a channel for injecting genetic material into the bacterial cell. It can be short, long, flexible, or rigid. After that, tail fiber. Tail fiber recognize and bind to the specific bacterial receptors and help the phage attach to the bacterial surface. And last is the base plate. Base plate is found at the end of the tail. This structure anchors the phage to the host bacterium. It also contains receptor binding proteins that help to recognize the specific host. In this slide, we can see the different types of bacteriophages. On the basis of their structure, bacteriophages are various types, such as filamentous type bacteriophage. First image shows the example of filamentous phage. Example include M13 bacteriophage, which is long, thin, and rod-shaped. Second image shows the example of tailed phage. It is most common type of phage. Its example include T4 bacteriophage. And third image is the example of icosahedral phage. It has spherical capsid without a tail. Its example include MS2 phage. In this slide, we will discuss about the life cycle of bacteriophages, that how bacteriophages replicate and produce their progeny. The life cycle of bacteriophages follow two main pathways, the lytic cycle and the lysogenic cycle. A phase that follows the lytic cycle is called virulent phase. This is called virulent phase because it immediately destroys the bacterial host after infection. And a phase that follows the lysogenic cycle is called temperate phase because it does not immediately destroy the host cell. Instead, it enters a dormant phase, allowing the bacterial cell to survive and replicate with the phage DNA and integrate it into its genome. First, we discuss about lytic cycle of the bacteriophage. The lytic cycle of bacteriophage have following steps: adsorption or attachment, penetration, DNA synthesis, assembly, and lysis. This is the cycle, lytic cycle of bacteriophage. First step is attachment. In first step, bacteriophage is bind to the receptor molecule on the plasma membrane of the host cell. And second step is the penetration. Bacteriophage contain enzyme lysozyme that drill a hole in bacterial wall and inject its genome into the bacterial cytoplasm. Bacteriophage done it by contracting its sheath. After that, replication. Bacteriophage replicates its genome and uses bacterium metabolic machinery to synthesize bacteriophage enzymes, and bacteriophage structural components. After that, next step is assembly. In assembly, newly synthesized viral components are assembled into complete virus particles. And last step is lysis or release. The host cell bursts, releasing new phages to infect other bacteria. And second cycle is lysogenic cycle. This cycle allows the phage to integrate into the host genome and remain dormant. Steps of lysogenic cycles are attachment, penetration, integration, dormancy, and induction. This is the lysogenic cycle of bacteriophages. First step, attachment and penetration, is similar to the lytic cycle. The phage attaches and injects its DNA into the host cell. Second step is integration. The phage DNA integrates into the bacterial chromosome and become a prophase. Third step is dormancy. The prophase remain inactive and replicate along with the bacterial genome during cell division. And last is induction. Under stress like UV light, chemicals, the prophase exits the bacterial genome and enter the lytic cycle. Application of bacteriophages. Bacteriophages have potential application, especially in the field of medicine, agriculture, biotechnology, food industry, and in pest control. Here we discuss application one by one. First is phage therapy. Bacteriophages are used as alternative to antibiotics. Bacteriophages can be used to treat bacterial infections, especially those caused by the antibiotic-resistant bacteria. Phage therapy involves using specific phages to target and kill harmful bacteria, offering a potential solution to the growing problem of antibiotic resistance. Before antibiotics were discovered, there was considerable research on bacteriophages as treatment for human bacterial disease. Second application is in field of food safety and preservation. Bacteriophage is used to control foodborne pathogens. Phages can be used to target and kill harmful bacteria in food products, reducing the risk of contamination with pathogens like Salmonella, E. coli, and Listeria. They offer a natural, eco-friendly method to preserve food and enhance safety. Bacteriophage is also used in food packaging. Phage-based coating or spray can be applied to food packaging to prevent the growth of pathogenic bacteria on food products. Bacteriophages also have vast application in the field of agriculture to control plant pathogens. Phage can help manage plant disease by targeting specific bacterial pathogens that affect crops. This reduces the need for chemical pesticides and promotes sustainable agriculture practices. Phages offer an eco-friendly alternative to chemical bactericides, and reducing environmental contaminations. Phages target only harmful bacteria. Phage therapy also used in veterinary sciences to control livestock disease. Phages can be used in livestock farming to control bacterial infections without harming the animals or environment. In poultry, bacteriophages effectively used against Salmonella, Campylobacter jejuni, and for cattle, Bacteriophages used against E. coli, Salmonella, and Clostridium. Bacteriophages also have great potential application in the field of biotechnology and genetic engineering. Phages used to genetic manipulation. Phages can be engineered to deliver genetic material into bacterial cells, aiding in development of recombinant proteins, vaccines, or gene therapy applications. Phages for combating global warming. Methane is the second more abundant greenhouse gas after carbon dioxide, produced by methanogenic microbiome of cattle raised for human consumption. Bacteriophages can be used to decrease methanogenic archaea population in cattle microbiome and decrease methane emission. Bacteriophages also have vast application in environmental sciences to overcome the problem of like treatment and bioremediations. Wastewater treatments. Phages can help in breaking down and removing harmful bacterial contaminants from wastewater, providing a natural way to purify water. Ganga has high concentration of natural bacteriophages so that the water of the Ganga river never gets spoiled. That's the reason. And bioremediations. Certain phages are being explored for their ability to target and degrade bacterial contaminants in polluted environments. Phage target and eliminate pathogenic bacteria in contaminated soil, water, and industrial waste. These potential uses make bacteriophages a valuable tool across multiple industries, especially as alternative to traditional antibiotics and chemical treatments. Thank you all for your time and attention. Hope you find this presentation insightful.